All right, let's go over chapter 7, the blood chapter. Okay, let me just get this up here. All right. Now, this overlaps quite a bit with lab, but in lecture we talked about a few other things. So let's just go over this. So first of all, what we were discussing are what's in blood. So we started off talking about the actual plasma, right? So as you know, that blood is not just, the liquid, there's the cells in there. But in the plasma itself, we have the main components. Of course, most of it is water, right? And then we have some other stuff, all right? So we can see gases, nutrients, different types of stuff. But in 105, we highlight the proteins. And in the proteins, we primarily have these three major classes. And the first one are the albumins. So remember in class, I was trying to review why this works. And the principle here is that the albumins do not go out of the bloodstream normally. So they help to maintain water balance because, as you know, by this point in the course, we talk about water following salt, but it will follow its concentration gradient, always go from high to low concentration. So the idea here is that these proteins cannot leave the bloodstream, and the albumins, they will be drawn, water will cause the albumins will draw the water into the bloodstream, and it's fighting osmosis, which is going to be pulling water out, right? So I know albumins are not salt, but the thing is they are a solute, and water is going to follow that. So there's a balance between what's happening with the water being pulled in because the albumins can't leave the bloodstream, and the water that's getting pulled out because it's following salt. And we'll talk a little bit more about this later on in the semester when we do circulation, but suffice to say, the albumins are going to help you maintain your water balance because you want to keep that fluid in the bloodstream. You don't want all that fluid going out, right? Because it's going to be drawn out because of the whole water following salt thing. But as you know, the blood must remain fluid, so we have to keep that water in there. And how we do that is with the albumins, right? Then the other protein that we talk about here is fibrinogen. I'm skipping globulins for a sec, is fibrinogen. And this is what we need to maintain blood clotting, all right? And I'm going to talk about fibrinogen again in a sec. We talked about it in lecture, but I'll go over it. But remember, fibrinogen is a soluble protein, and that one is sitting there ready to go if you need to form a blood clot. We don't want the cells to form fibrinogen and then fibrin, which is what you need for clotting. We have the fibrin in a soluble form, which is fibrinogen, and that is ready to go for blood clotting. Right, because clotting has to take place very quickly. We don't have time to wait for proteins to be made. So it's already sitting there ready to go. Now the globulins, there are actually several kinds. And the one we focus on in chapter 7 are, of course, the antibodies, the gamma globulins, which I've listed here at the bottom. So we talk a little bit about those, and I will go over that again. But there are another two types of globulins in the blood, the alpha globulins and the beta globulins. We're going to talk about them in 105, but I just want you to know that they're there. There's not just one kind of globulin. We mostly talk about, like I said, the antibodies. These alpha globulins, they're regulatory proteins, and the beta globulins, you might know them better as what they associate with. So low density lipoprotein and high density lipoprotein are good and bad, or bad and good fats. The LDL is what you call bad fat, and the HDL is the good one, but they're associated with proteins, and those are called the beta globulins, right? So, but for 105, we'll just concentrate on the gamma globulins, or the antibodies, right? Now, I'm just looking quickly at my notes. All right, now the other part of blood, of course, are the cells, or the formed elements, okay? Because they're not all cells, but globally we call them formed elements. So here we have three different classes. The first one are the platelets, and I'm going to talk about those, but remember we need those for blood clotting. Then we have the white blood cells, and remember the science term for white blood cell is leukocyte, the one with the L-E-U, right? Leukocyte. Those white blood cells come in 5 flavors. They come in the form of neutrophils. They come in the form of lymphocytes, monocytes, eosinophils, and basophils. And I'm going to go over each of those again, all right? And then we also have the red blood cells. And remember, the science term for red blood cells is erythrocyte, all right? So in this class, you've got to know, when I say leukocyte, that I'm talking about that whole class of 5 different cell types, all right? It's not just one cell type. Very often students in 105 get the term leukocyte and lymphocyte mixed up, okay? Remember, leukocytes are those 5 classes of white blood cells. Why are they white? Well, because they're not red cells, right? So the red cells, remember, their sciencey term is erythrocyte, okay? So if I say erythrocyte, you should know that that is a red blood cell, right? And I will talk about those in a sec. Now, what we can do with blood is we can spin it down. And when we spin it down, we get a layer of plasma, all right? And you can see 55% of that, and that varies. We'll talk about that in a few minutes again, but basically 55% is plasma, 45%, and these are the heaviest part, that's going to be at the bottom. You know, the centrifuge things, the heavier things go to the bottom, and those would be your red blood cells. And if you looked at these in class, in lab, and you think, oh my God, they're so small compared to the white cells, which sit here in this buffy coat, all right? So you think that these cells are bigger, they should be at the bottom, but remember, those red cells contain iron, right? They contain iron in the hemoglobin, and that's what makes them heavy. So they're down at the bottom, okay? So 45% is going to be about 45% is your red cells, 55% is your plasma. In the middle, you're going to get your buffy coat, and that's with your white blood cells and your platelets, all right? Now, next idea. All right, now with your leukocytes, your white blood cells, in general, what do they do, right, in general? Well, you can think of them as defending the body, right? They all have a role to play in this, and we will go over that in great detail. But one thing that they have in common is that all of the leukocytes will have a nucleus, right? The nuclei will have different shapes and different other aspects, and sometimes they have these little granules, but we will talk about those in a few minutes as to what makes them separate, all right? But in general, they all have a nucleus, and they also all have mitochondria, all right? So we'll talk about that in a bit. The other thing that I mentioned in class is how we develop these blood cells. These blood cells are all formed from a common stem cell, and we say it's a pluripotent stem cell because it forms different types of cells. It's not a totipotent stem cell. That's what we have when we're developing developing humans, and you can form any kind of tissue. Pluripotent can form more than one cell type, but as you can see here, they're only forming blood cells. They can be teased into forming different types of cells in different ways. We're trying to do that without having to use embryonic stem cells. That's the other term for totipotent. Those totipotent ones will form all the cells in the body, all right? But you have a pluripotent stem cell in your bone marrow, which has the capability of forming all of the different blood cell types. And in 105, we're not expecting you to learn all of this. But we talk about the formation of the red cells or the erythrocytes. But I want you to sort of just visualize that this is all of these different cell types are coming from a single type of stem cell, all right? And I mentioned in class that there's 2 sort of branches to these cells. So they're either going to be lymphoids, or they're going to form your different lymphocytes and your natural killer cells, which we don't talk about in this class, but you might have heard of them before. Or they're going to form these things that are called myeloid cells, which are basically all the other cells, all right? If you go on and do A&P, you'll learn a little bit about immunology. And you'll see that these are the ones that are doing your what we call educated immune system, where they respond to vaccination, right? Whereas these ones are protecting you and they don't require vaccination. That's a different kind of protection that they'll offer, but we'll talk about that in a few minutes. But remember, your cells are coming from a single type of stem cell, and they either go myeloid or lymphoid. The lymphoid cells don't have granules, so sometimes they're called agranular. And the myeloid cells, very often they have these granules, so sometimes they're called granular. All right, these are just different terms that you'll see when you're reading up on this, or if you do the Google. But I wanted you to get an appreciation of what's happening here. All right, now we're going to focus on red cells, or the erythrocytes, in this chapter. So I'm going to start off talking about those and the different types of aspects of them and why they're so important. I mean, obviously, they're red, so you know you've got a lot of them because your blood is red, especially when it hits that oxygen. And why is that? So let's go over that. Right. So the first type are erythrocytes, like I said, also called your red blood cells. They're responsible for the carrying of gases. You probably know, if you know anything about blood, that they're responsible for carrying oxygen, but that isn't all they carry. And we talk about this later on in the course, so I'm sort of giving you a primer here. Erythrocytes, of course, they carry oxygen. How do they carry that oxygen? Well, they carry it on a protein called hemoglobin, right? And we've talked a little bit about hemoglobin earlier in the course when we talked about protein formation. This is a protein that's made up of 4 subunits, and each subunit comes together to form the hemoglobin molecule. And in each of those subunits, there's a structure here, a porphyrin structure, and it is a heme structure, we call it, and that's where we get hemoglobin. It's a globular protein, and it's got heme in it. And in the middle of each heme molecule, that's for each of the subunits, there's an iron. And as you know, iron attracts oxygen. And you know what happens to your car when that iron is attracting that oxygen? It rusts out and it turns that reddish color. And that's why when red blood cells are carrying a lot of oxygen, they'll appear red, because that iron, that F2, iron 2 there, turns that sort of reddish color, and that's what's responsible for that. And that's why your deoxygenated blood, so if you look at your blood vessels, some of them will look a little bit blue. That's where you've already taken out that oxygen, your tissues have removed the oxygen, and that's going to be your veins flowing back to your heart, okay? But we'll talk about that later on, right? Now, what else? I said to you that we're not just carrying oxygen. Well, turns out that heme will also bind to carbon dioxide. All right, we'll talk a little bit more about that later on in the course, but we've got different names for hemoglobin depending on what it's carrying. So when hemoglobin in your red blood cells is carrying oxygen, it's called oxyhemoglobin. When that oxygen comes off, it's called deoxyhemoglobin. Now, when it is carrying CO2, right? When your hemoglobin is carrying CO2, and we'll talk about later on in the course how this works, but when it's carrying CO2, it's called carbaminohemoglobin, all right? So that's this one here, all right? And then we have this one, carboxyhemoglobin. Now, this one's dangerous, all right? So what is it carrying? Well, in this case, what it's carrying is carbon monoxide, right? Now, it turns out that hemoglobin has a higher ability to carry carbon monoxide than it does oxygen. Now, this is why carbon monoxide is so dangerous, and that's why they don't want you running your generator in your house or starting your car with your garage door closed, because what's being produced is a lot of carbon monoxide, and it's a toxin. Why is it toxic? Well, it will take the place of oxygen on your hemoglobin. So what will happen, and I've actually had students in my class that have suffered through this. Thankfully, they managed, somebody caught them before they passed away. But what will happen is that carbon monoxide will bump off your oxygen, and the oxygen will not be able to rebind because carbon monoxide is binding more tightly to your hemoglobin. So basically, you pass out, and eventually you asphyxiate or you suffocate because you're not getting enough oxygen to your tissues. This is why it's so dangerous, because you can't smell carbon monoxide. So very often someone will just feel sleepy and pass out, and then if nobody finds them, they will die of carbon monoxide poisoning, and that's how it works. It replaces your oxygen. So suffice to say, hemoglobin carrying carbon monoxide is called carboxyhemoglobin. All right, so those are the 4 forms that we talk about in this class, and try to keep those straight. And we will talk a lot more about carbaminohemoglobin a little bit later in the course, all right? But understand that hemoglobin and your red blood cells will carry all of these gases. So oxygen, carbon dioxide, and carbon monoxide, all right? Now, different aspects about erythrocytes that make them special. Well, they don't have a nucleus. And the other thing is that they don't have mitochondria. During development, they will have these things, but a fully formed erythrocyte will not have them. So let's think about why that is. And I went over this in class. Now, first of all, if you don't have a nucleus, the cell will have this, sort of donut kind of shape to it. And this is really good for gas exchange because the cells are very, very thin. And remember, this is going just by diffusion. So if you have this sort of fat cell here, it's going to be really inefficient for the gas exchange. And erythrocytes have to be really good at gas exchange, so losing that nucleus is good. The other thing is that you don't want your erythrocytes to be using energy making proteins. Now, think about that. Energy, what you're using energy to make proteins, and if you don't have a nucleus, you're not going to be making proteins as actively. There are some that are going to be made in the erythrocyte, but you don't want them actively making all kinds of things, because you're going to use up energy. If you're going to use up energy, you're going to use up our energy carrier, our ATP, okay? And we don't want to do that, all right? So we don't want to be using up oxygen, which is the other reason why we don't have mitochondria, right? So, because as you know, in order to make the most ATP possible, we use oxygen. And it would really defeat the purpose of an erythrocyte if they were actively using oxygen to make ATP to run the cell, if their prime job is to carry oxygen to your tissues. So this is one of the really great benefits of having no mitochondria and no nucleus. So you can see that the erythrocyte has evolved specifically for its job. We don't want to be using up the gases or oxygen when we need it delivered to our tissues. All right, so these are the 2 really special things about the erythrocytes and why they look the way they do. All right. Okay, so let me just see. Oh, that's the other thing. In my notes on page 3, I said number 2, must be able to get into the smallest vessels. So that's the other good thing about not having a nucleus, is that these erythrocytes are very flexible. They don't have that big old nucleus in the middle making them sort of clunky. So they're really able to do this. And if you remember, I showed you that video in class of the movement of blood. So it's passing through the tissue so quickly, and it can easily get damaged if they had a nucleus in there. Okay, great. All right, so now talking about the formation of these erythrocytes. Now, we use up erythrocytes very quickly. They get damaged and there's a really, really high turnover. And we talked about that turnover in lab. Okay, I think it was like 2 million a second or something like that we turn over. So we need to be able to make these efficiently. And we talk a little bit about what we need to make the red blood cells, all right? So here you can see, if we look at the steps involved, you can see that we're starting with something we call the pro-erythroblast. Every time you see blast in biology, that means something early. So this is sort of an early type of cell. And then we have these things called reticulocytes. So basically a reticulocyte is a mature erythrocyte, but it still has its nucleus. So you might see some of these in the bloodstream. They can still carry some oxygen, but not very well, because it's still got that nucleus in there. But these retics, so they'll do a retic count on your blood as well, but very often these stay in the marrow, but some will get out. But these are the reticulocytes. They still have a nucleus. Once the nucleus is ejected, it can then form a mature red blood cell or erythrocyte, right? Now, how does that stem cell know that it should do this? All right, so I'm going to talk about this. So clearly, we make the red blood cells in the bone marrow, right? Remember, in our red marrow, that's where we're making all of these blood cells. They go out into the circulation. They last about 120 days, okay? After which time they're broken down, or they start getting collected largely in the liver and the spleen. Now, the reason those two organs, and I think I mentioned this earlier in the course, but the liver and the spleen are very red. So if you would open any mammal, you would look in there and you'd see a very red organ, very, very big one, that's the liver, and then a smaller one, which is the spleen. But the reason they're so red is because here is where we're having the turnover of the iron and the red blood cells. And the other reason is because blood circulates through there differently. We'll talk a little bit later about the different types of capillaries. But here, these two organs really are bathed in blood, so that's the other reason that they are very red. But what's going to happen here is that the old red blood cells are going to be phagocytosed by macrophage, and they're going to be broken down, and we'll recycle some components. All right, so for example, the iron very often is recycled, so it's going to be carried by proteins in the blood back to the red marrow so that it can be used to make new hemoglobin to be put into the red blood cells. Other things, the heme molecule itself is broken down and eliminated, and it has color to it because it has a ring structure. Ring structures very often will provide color, and that will color the feces and the urine, all right? And that's sort of what I'm getting at here. So erythrocytes themselves have a short life cycle. The macrophage are going to break them down, and some components are going to be reused and some are going to be removed, all right? Now, the next idea, how does the body know, or how does that stem cell know that we need more red blood cells, all right? Because sometimes during life, we're going to need more than others, and we'll talk about a few examples of that later. But there has to be a signal to that stem cell. So remember, the stem cell can form all of these different cell types. So there has to be some sort of signal that tells that stem cell, form red blood cells. We need more red blood cells. And what that signal is in the body is the amount of oxygen in the blood, right? And it works through the classic negative feedback system. So we have a certain amount of oxygen carrying capacity that we need in the blood, and that is our set point. And if our oxygen levels drop, there are cells within the kidney, all right, that are sensitive to the amount of oxygen that we have in the blood. And when those levels fall, those cells in the kidney are going to release a hormone. And we're going to talk a bit about this hormone later. This is one of the many hormones that you're supposed to learn in this course, but it's called erythropoietin, or EPO for short, okay? So those oxygen-sensitive cells in the kidney, if they sense that the oxygen levels in the blood have dropped, or we don't have enough red blood cells, that's what that translates to, they will release the hormone erythropoietin, and it will travel through the blood to the red marrow, and that's going to stimulate the production of erythrocytes. And so then the stem cell is going to form those red blood cells. They will be released into the blood. Then what's going to happen is our oxygen carrying capacity of the blood will go up. You'll get back to the set point, and then those cells in the kidneys will stop producing erythropoietin because we're back up to where we're supposed to be. All right. So that's how that works. So we have this negative feedback loop, and the key is erythropoietin being released by these cells in the kidneys. All right. Now, So here we have two things. So we know that in order to get erythrocytes to be produced, we need EPO. We definitely need that. The other thing, though, of course, is we need iron, because the iron is what is going to carry that oxygen when it's in the hemoglobin molecule, right? So if we're low in iron, even if we're making more red cells, we're not going to be making enough hemoglobin. So we need to have both. So we need erythropoietin as the signaling molecule, and we need iron, all right? Now, this brings up the idea of blood doping. Now, what does that mean? All right, so doped blood, when it comes to athletics, has meant that you have somehow increased your oxygen carrying capacity artificially. And that's cheating, all right? So there's two different ways in which people have cheated to do this. One way, okay, is to just increase the number of red blood cells, all right? So normal blood cells, normal blood is going to have 40 to 50% red blood cells, like we had already talked about. And doped blood, one way to do this is athletes will sort of take some of their own blood and bank it. So they'll store up their own red cells, and then before an event, they will have someone inject those red cells back into their blood. So in that way, they're sort of artificially using their own red cells, or they could get a donor. Now, this is really, really dangerous, all right? Because what happens is when you increase the number of those cells, your blood gets thicker. And when it gets thicker, it gets more difficult for the heart to pump it, all right? So this is not a good thing to do. And it's pretty easy to catch this, all right? The other thing that athletes have been known to do is also take injections of EPO. So they'll take an injection of erythropoietin, and that will stimulate their stem cells to make more red blood cells in that way. So without using a donor or banking their own blood, they'll just increase the red cells, all right? Now, why does an athlete want to do this? Well, if you've got more red cells, you can carry more oxygen. If you can carry more oxygen, in theory, you can make more ATP. You can make more ATP. That means you can have your muscles working longer and harder for longer periods of time. And that's sort of the incentive to do that. All right. I mentioned in class one way to sort of go around this and sort of cheat, right? So a lot of athletes, they'll do something called altitude training. So what happens is that at higher altitude, so up in the mountains, there's lower oxygen level in the atmosphere. So your body will naturally, with time, make more red blood cells without, doing any of this doping business. So I before the last Olympics, was watching this one marathon runner. I can't remember his name, but he was really good. Anyways, he was training in the mountains, and that way he was increasing his oxygen carrying capacity in that way. And then when he went to the Olympics at Rio, and of course that's down at sea level, so he was able to very efficiently carry that oxygen. So that's a way of going around it. Without, doping in this sort of unnatural way, right? Okay, now, how do we measure? How do we know? How do we know how much, how many red blood cells you have? And how do we know if you've been cheating? Other than looking for levels of EPO in the blood. Well, a way to do this is measuring the hematocrit. And everyone's got a hematocrit, and it's based on spinning the blood, right? So this goes back to what I was saying at the beginning here. We can spin blood down, and we get 45% about being your red blood cells. Well, that's your hematocrit, or your crit. So it'd be 0.45. So that's just taking the percentage without moving it over the two decimal places, right? So here you can see, and that's why it's actually drawn like this, as if it was spun down. And notice it's also called the packed cell volume. If you see a chart in the hospital or your own chart, I don't know if the doctors actually has that in your, your chart in the office or not, but you'll see the hematocrit or the crit. And that is saying, if you spun the blood down, what percentage or what proportion of the blood is going to be your red blood cells. And you can see there's actually differences, so that that 45% is sort of an average. But so if you look at the female, a normal hematocrit is about 36 to 46. And for a male, it's 41 to 53. And a newborn, it's really high, right? 49 to 61, all right? And there's a lot of reasons why it varies that much. I mean, you can think about it. If you're someone who lives at sea level versus someone who lives at the top of the mountains, you're going to have very different crits. And also for women, it will change through the cycle, for example. So there's reasons that they'll vary, but they sort of average it out at 45 to 46%, right? And it is a measurement. All right, notice this is in red. The hematocrit is a measurement of the blood's oxygen carrying capacity, because your ability to carry oxygen in your blood is reflected by how many red cells you have, assuming that you have enough iron. All right, so this is used as the measurement of your oxygen carrying capacity. All right, so hopefully that makes sense. Now, anemia. So if your reduction, or you have a reduction in your oxygen carrying capacity, you're anemic, all right? And there could be a variety of reasons for this, and I've listed them here. I'm not going to get you to memorize all of these, but this is a very common thing. So how do you know you're anemic? Well, there's a whole bunch of possible symptoms up above. And I was actually just talking with someone who told me he was traveling unexpectedly or uncharacteristically in Colorado, and he very quickly drove his car up to some mountain peak. I can't remember how high he said he was, but he was really up there, and he went very quickly with his car. And he said when he got to the top, he felt really nauseous and dizzy. So he was suffering from anemia because he wasn't being able to carry enough oxygen, because he's normally down, at a lower altitude. And he said he really felt that. But anyway, so what are the different kinds? So we have iron deficiency. So here, if you have low iron content, either because of your diet, or some people just have a poor ability to absorb it, you're going to have less hemoglobin, and you're going to have less oxygen. Hemorrhagic, that's due to blood loss, right? Usually due to an injury, but it could be because of menstruation, ulcers. Another one is parasites. That sounds really gruesome, and we don't usually have that in North America, but that is a factor because parasites also want that iron. And they'll also cause bleeding. So hemorrhagic anemia. Then there's pernicious, and this is due to low vitamin B12. Now, why is this? I didn't really talk about this, but you need B12 to make hemoglobin as well. So you also need it to make efficient erythrocytes. So if you're low in B12, you're also going to be anemic. So hemolytic, this is due to the rupture or destruction of erythrocytes. There could be a variety of reasons for this, but this is often associated with another anemia, sickle cell anemia, which we'll talk about in a sec. Due to renal failure, don't forget, in order to get red blood cells, we need iron, but we also need erythropoietin to signal production. And if you have a problem with your kidneys, if you're having renal failure, you're not going to be making EPO. So that's another reason to be anemic. And then, of course, sickle cell. Sickle cell anemia is genetic. Now, some of you know about this. I usually have some students in class that have this or are carriers, all right, simply because of the mix of backgrounds that we have at Marsville. Now, what is this? Now, what can happen is that there's a genetic mutation. It's a point mutation, so there's a one change in the DNA, so you can see it here. So and we'll talk about this later on in the semester, but in order to get an amino acid, you need a code of 3 nucleotides in your DNA. And normal hemoglobin will have this amino acid. It's called glutamate, and it's coded for by a GAA. And if that switches, okay, and you get a GUA in your mRNA, what you're going to code for instead is a valine. And Glutamate and a valine have very different properties. You don't need to know this for 105, but for example, a glutamate is a charged amino acid, which means it likes to be on the outside of a protein. Valine is not charged, so it likes to be on the inside of a protein. So what ends up happening is that one amino acid change causes the folding of the protein to be completely different. So it's no longer globular and isolated on its own. What it tends to do then is it forms, the proteins will join and form these strings or fibers. And this will cause the red blood cells to change shape. So normally they have these beautiful donut shapes. And then with someone who has sickle cell, what will happen is that these fibers will form, and it will cause the red blood cells to form a sickle shape. And I know this day and age, people don't know what a sickle is, but it used to be a long sort of C-shaped blade that they used to use for harvesting wheat, for example. And that's where it got this name, sickle cell, right? So you can see the change in the shape from a normal cell to a sickled cell, all right? And these cells, first of all, they don't travel well in the bloodstream. They cause bleeding because they have this sort of sharp shape to them. They'll cause damage to the blood vessels, and they don't carry oxygen very well. So this causes a lot of problems with someone who has sickle cell anemia. All right. All right, yeah, there's other things I wanted to tell you about this. But if you're really interested in this, remember we have a class blog, and past students in other semesters wrote about different aspects of sickle cell anemia. So you can check out the course blog and read up on what some of your previous colleagues or classmates had wrote about in terms of sickle cell, all right? Okay, so that's our erythrocytes. So there's a lot we cover with erythrocytes, and you should know a lot of that, all right? Now, with leukocytes, remember, this is your 5 classes of white blood cells. And remember, sort of an overall theme that these cells do is sort of help defend you in different ways, okay? And with our white blood cells, we have sort of 2 main flavors. We have the granular ones, meaning that they have granules, and then the agranular ones, meaning they don't have granules, or at least not granules you can see. So within the granular ones, we have the neutrophils, the eosinophils, and the basophils. And the agranular ones, we have monocytes and we have lymphocytes, okay? But remember, all 5 are leukocytes, or white blood cells. Some of those are phagocytic, all right? Some of them have the ability to phagocytose things. And of those, we have the neutrophils and the eosinophils and the monocytes. Monocytes, those are in the blood. And when they leave the bloodstream, which I'm going to mention again in a few minutes, they become macrophage. So, and those cells are dedicated to being phagocytic, all right? All right, now, let's talk about these a little bit. Now, I showed you this video in class. I strongly encourage you to watch it if you missed class. So click on the link, watch this video. It's really important because I want you to understand that these white blood cells, the leukocytes, they don't just exist in the blood. They leave the bloodstream and do their jobs. And all of them, except for the monocytes, go back to the bloodstream later on, okay? Remember, the monocytes, they don't go back because they become macrophage, right? So this is why almost every tissue we talk about, or organ system we talk about, we have macrophage and they have a particular name. And those used to be monocytes that left the bloodstream and then just stayed in the organ. But the other white blood cells, the basophils, neutrophils, eosinophils, and lymphocytes, they all can go back into the bloodstream, all right? But in order to leave, there's a process. And you don't need to learn the process. I sort of talk about it in class because I used to study this for my master's and for my PhD when I went in terms of tumor cells, because tumor cells also do this. So I have a bit of a background in this, but I kind of just wanted you to know that there is a process involved. They don't just exist in the bloodstream and then find themselves in the tissue. And even though it looks like that in the video because it happens so fast, there is a whole team of scientists and labs throughout the world that are studying how this happens, all right, for a variety of reasons. And this sort of shows you how it happens. And when the cells do go leave the bloodstream, they can either go through the endothelial cells making up the blood vessels, or they can go between them. All right, so this is really, really cool. And you can sort of see white blood cells leaving the bloodstream if you watch this video, okay? So remember, all leukocytes leave the blood and circulate in the tissues to help protect the body in their own specific ways, okay? And that process of leaving is called extravasation or diapedesis, right? Okay, now let's look at the granular cells. So here we have the neutrophils, eosinophils, and basophils. And remember, in lab, you must be able to identify these, okay? Now, neutrophils, how can you spot those? Well, they don't have granules that will tell you, but their nucleus is usually in several lobes, all right? You'll see that you'll talk more about it in lab, but that's the telltale sign. If you see that's a cell that looks like that, you know it is a neutrophil. So the granules are not red or blue. They have granules, but they don't stain red or blue with right stain. They are phagocytic, and they are phagocytose bacteria and fungi. And you'll have increased numbers of these in a bacterial infection. Eosinophils, their granules look red. Okay, so you can see the nucleus still looks like that, but they have granules that you can see. They're red, and they moderate allergic reactions. Their numbers will go up if you have a parasite. Okay, so how does that work? Well, they're not big enough to phagocytose a whole parasite, but these granules have enzymes that can break down a parasite, and then they can phagocytose little chunks of the parasite. Okay, so there's going to be many, many eosinophils working together to help fight a parasite. Basophils, now their granules are blue. Okay, so if you know, most students these days don't learn Latin, and I definitely did not learn Latin, but I picked up some as I went through my scientific education. But eosin usually means red, and baso is implying blue, so that's where we get those names, okay? So the basophil, the granules are blue, and those granules release histamine, okay, during inflammation. And we need that inflammation, even though you might hate it when you're having an allergic reaction, all right, because it's making your eyes all watery and you swell up and everything. But it brings about healing, all right? So this is sort of why we have this. We'll talk a little bit about it later when we talk about circulation. But we need this to bring up inflammation. And notice that the eosinophils are going to sort of moderate this. So they kind of work to dampen it a little bit, but you do need to have that inflammation, all right? So there's sort of, basophils are doing their job when you're having an allergy, all right? Okay, hopefully I'm not missing anything here. Okay, behind in my notes, I'm just going by what I know. Okay, I have this note on the basophils in my notes. Why do we want this inflammation in the first place? What it's doing is that it's bringing about nutrition, to help heal. So it's going to make the blood vessels leaky, so nutrients are going to come out. It's also going to make it easier for cells to come out that are going to fight an infection, so a neutrophil or an eosinophil. And there are these biochemicals that you need, and they're also going to be recruited during these reactions, all right? All right, now, agranular leukocytes. So here we have the monocytes, which I mentioned to you, become macrophage after they leave the blood, all right? So they're going to be activated in long-term infections against viruses and bacteria. And then we have the lymphocytes, and we really don't talk about these in this class because we skip over our chapter on immunity, and so we don't really talk about it. But this is your learned immunity or educated immunity. These lymphocytes are your antibody producers. They're going to be either B cells or T cells, and they're going to fight many types of infections, and specifically the ones that are making and releasing the antibody are these certain type of B cells that become plasma cells. And you can see they look very different than monocytes in that the monocyte you can see this light blue here is the cytoplasm, and they have this big old nucleus here, whereas the lymphocytes pretty much just look like a big old nucleus, and you don't see very much of a cytoplasm, all right? So that's how you can tell those two apart, all right? Now, Moving on. All right. So when they're trying to figure out what's wrong with you, if you're not feeling well, they often can look at the numbers of those white blood cells to get an indication, right? So for example, if you have any high white blood cell count, we'll say that you have leukocytosis, all right? It indicates either an infection or a leukemia of some kind, right? So for example, here you can see there's a lymphocytic leukemia. We've got way more lymphocytes than normal. And I know if you looked at your blood smears in lab, you did not see this many lymphocytes, so this would be indicative of leukemia, right? Then if you have very low white blood cell count, all right, so that's called leukopenia, and that usually indicates some sort of catastrophic disease such as AIDS or bone marrow failure, sometimes severe malnutrition, and clearly chemo, that would do it as well. Now, the other thing is say you have a lot of eosinophils, for example. Let's go back. If you have a lot of eosinophils, well, that might indicate that you have a parasitic infection. Or if you have a lot of neutrophils, well, that might indicate some sort of bacterial infection. So that's the other thing that they'll look at, all right? So they can use certain instruments to allow them to count how many cells or what percentage of your white blood cells are these different types, and that will help, however it's deviating from normal, help to assess what is wrong with you, okay? Now, this little thing on leukemia, what is it? There's two different types, acute and chronic. So you've probably heard of this. Acute just means it's a really rapid onset. Chronic means it's slow, all right? Now, I put this little thing, you don't need to know this for your exam, but I'm just doing this for your information. So these acute lymphoblastic is going to be of the lymphoid cells, right? And myelogenic, so that's going to be of the non-lymphoid cells. So those are the two different types. And very often acute cancers show up more in children than they do in adults, right? And that's not a hard and fast rule, but very often if it's a child who has leukemia, it's going to be an acute type. Another way of them knowing that there's a leukemia happening is that cells will be released before they're from the red marrow before they mature. All right, so that's just a little bit of the differences between these, because most of us know someone who's had leukemia or they've heard of someone with these, and these terms are kind of confusing. So the point of this is just to sort of help you sort of digest what these mean, okay? Okay, now let's move on. Now the next idea are things that can go wrong, and a lot of these are going to be covered, or depending on when you're listening to this, or where covered by some of your classmates, all right? But we have multiple myeloma. This is a cancer of plasma cells, all right? So these are the type of B cells that produce antibodies. These ones don't normally leave the bloodstream, the bone marrow, and enter the bloodstream, but what they'll do is they'll sit in the marrow, and they cause a lot of the bone to sort of get very brittle and painful. And they don't produce very good antibodies. They don't work very well. So very often, someone who has multiple myeloma, they'll know this because the bones feel very painful and tender. And because the bone is breaking down, the blood calcium levels are going to be high, all right? Then we have mononucleosis. Now, this is caused by a viral infection of the lymphocytes, okay? It's an EBV infection, Epstein-Barr virus, swollen tonsils, all right? Now, why is it called mononucleosis when it's an infection of the lymphocytes? Now, if you go back to our pictures of the monocyte and the lymphocytes, it's a reason I put them together. So here, notice the monocyte and the lymphocyte. They look very similar. Now, remember, mono is a viral infection of the lymphocytes. So what ends up happening is that these lymphocytes get so full of virus that they actually swell up and start looking like a monocyte. All right, so that's why they call it mononucleosis. So don't let that confuse you. So mononucleosis is an infection of the lymphocytes. It will cause them to look like monocytes. All right, so that's what's happening there. Um, Now, before we get into the next thing, let's talk about platelets. All right, so platelets, they're a formed element on their own. They are not a full cell. They are fragments of cells. All right, they don't have a nucleus. All right, um, but they're very powerful, and we need them for forming blood clots. All right. They are fragments of cells that reside in the bone marrow called megakaryocytes. So the megakaryocyte will sort of extend a part of its membrane into the blood vessel, and it will release portions of it, and that is the platelets, right? And in your blood smears, sometimes students will confuse this for a basophil because it looks like a bunch of purple stuff. But it's not. It's just fragments of megakaryocytes. All right. So here, what do you need to know? Well, platelets are needed for forming blood clots or for hemostasis. All right. So let's talk about the steps of hemostasis. All right. So blood clotting has three main steps. All right. So the first thing is you're going to have some sort of damage to the blood vessel. Now, the first thing that the body's going to do is try to restrict the blood flow because we don't want to be bleeding out. So what's going to happen is that the smooth muscle surrounding the blood vessels is going to spasm or tighten. All right, so the first step is vascular spasm, and that is to try and slow the blood flow through that area where there has been some damage or a breach. Okay. Now, the second step is that the platelets are going to come here and they're going to get stuck. And there's a variety of reasons that this happens, right? We don't talk about it in this class, but there's sort of, exposure to proteins on the outside of your blood vessels that normally blood cells are not exposed to because they're on the inside of the blood vessel. So that sets out warning bells in a way. And the platelets go, oh my gosh, I can see all these extra proteins. And that sort of recruits them to form this plug. All right, that's sort of a very simple way of explaining it. So they start sticking to each other. Why did they start sticking to each other? Well, they start to get all bumpy, all right, and they start holding on to each other, whereas normally they don't. Okay, so second step, platelet plug formation. And then the last step is the conversion of fibrinogen. Remember, that's our soluble protein into the insoluble form called fibrin. And this makes a mesh, and it traps and cross-links those platelets. It's going to trap within it some red blood cells because it happens really quickly, and that's why blood clots, if you have a scab, for example, this will happen in the body too, but if you have a scab, that's why it has that red sort of blood, because there is blood stuck in that mesh of fibers, okay? And that mesh of fibers is called fibrin, and that's formed from your fibrinogen, right? So if you've ever had a scab and you kind of, it's really tight at first and it's holding onto your skin, well, that's because of that mesh of fibers that's sort of holding it in place, okay? So those are your three steps of blood clotting. Blood clotting is called hemostasis, and the first step is vascular spasm. Second step is platelet plug formation, and the last step is the clot, right? So you have to know what's happening there, and you need to know the order. And by this point in the semester, you know I love sequences. So always when you see a sequence, you got to learn it, all right? So here's your sequence for this chapter, okay? All right. I'll just quickly look at my notes. Is there something I'm forgetting? No. Okay, so how does this happen? All right, how do you get from a soluble protein to an insoluble fiber? All right, now the other thing in this course that you should remember is that enzymes, which are also proteins, but they're responsible for almost every reaction in the body. All right, so not only do you have the soluble fibrinogen circulating in your blood, but you also have an enzyme that's called thrombin. And that thrombin is the enzyme that's going to convert fibrinogen into fibrin. But you don't want that to be active either, all right? Because you only want it to be activated when it needs to be. So it's circulating in a form called prothrombin, all right? So I know that sounds really confusing, but this last step that I'm showing you here, the conversion of fibrinogen to fibrin using the enzyme called thrombin, which is an active form of prothrombin, okay? That's activated by an activator called prothrombin activator. That is only the last step in like an 18-step program to make this happen. All right. So clearly you don't want blood clotting happening when you don't want it to, or when you don't need it, because it's really bad for you. But since you need it very desperately to happen very quickly, there is a whole bunch of players that are all ready to go. Okay. And you might be aware of a disease called hemophilia. That's a genetic disease. And people who have hemophilia, there's a couple of different types, but there's one main one, and that needs one of these factors in the clotting to be activated. It's called factor 8. And they have, they're compromised in one of these upper level steps in the formation of clot formation, right? So if you take A and P, you're gonna see more of this pathway, all right? But what do you need to know? Well, you need to know in 105 that in order for the last step of hemostasis to take place, you need fibrinogen, the soluble form, to be converted into fibrin fibers, and that is done using an enzyme called thrombin. And that enzyme is circulating in the blood, also in an inactive form called prothrombin. And it's going to be activated if you need to form a clot, okay? And there's many, many steps involved in the formation of a clot, but this is just the last one. Okay, so as I said here, many of these important enzymes are pre-made in an inactive form so that they can quickly be activated when needed. So clotting has to take place quickly. So this includes the enzyme thrombin. That enzyme is going to be converted from prothrombin, and it's going to do its job converting fibrinogen to fibrin. All right, so here, not that you need to learn all this, but this shows a little bit more of the pathway. And notice the other thing that seems to be really important in the formation of clots is calcium. All right, so we don't really talk about that in terms of you know, why we need it, but calcium is also really important for forming clots. Another thing that we need is vitamin K, and we're going to talk about that later on in the semester when we do digestion. All right, so a lot of these activators are based on vitamin K, and so we'll talk about that later. Okay, make sure you know the stuff in this red box for the exam. All right, okay. Another thing that can go wrong: thrombocytopenia. This is if you have a reduced platelet number. So what happens then is that you have very easy bruising and bleeding, and that can be caused by a variety of things. All right, so anemia, viral infections, radiation, leukemia, blood disorders, but suffice to say, you've got a reduced platelet count, all right? Then another one is blood poisoning. We didn't really talk about that. What is it? Well, it's a bacterial infection of the blood. So what's happening is that you've just got so many bacteria, you're overwhelming the immune system, and that causes blood poisoning or septicemia. And you'll see it you can see red streaks. It's basically inflammation in your blood vessels. So you can see that on the skin near an infection. All right, so that's what's happening there. All right. Now, in terms of what I really need to review with you again is your blood types. All right, so since we're doing the blood chapter, we need to understand blood types. All right, and you really need to wrap your head around this. This is a big thing in 105, all right? So if you're having trouble, make sure you come and see me. I'm always your best resource, so please, please, please come and see me, all right? And please ask questions, all right? Now, let's go over this. Now, why do we care? So first of all, why do we care? Well, because if you're not given the right blood in a transfusion, you're gonna have a rejection of that blood. So how does that work? Right? So the idea here is that if we start from first principles, every cell in the body has proteins on it, okay? And those proteins are called antigens. Why are they called antigens? Well, because antibodies can recognize them. And these are very specific for different cells. Here I'm showing you a bunch of antigens on a bacterial cell, right? And indeed, you know that if you get vaccinated, you're going to start building antibodies that are going to recognize that infection, right? So whether it be bacterial, right? That's what your antibodies are recognizing, right? And it's those antibodies are recognizing those proteins that are found in a bacterial cell. But understand that even all of your own human cells have proteins. And I said in my lab, I said, okay, if I needed a new kidney, could Dr. Crawford give me a kidney? And most people will say, well, probably not. And I said, well, why? Well, because she might not be a match. Well, what do we mean by that match? Well, that match means is that she needs to have similar proteins on her kidney that I have so that my body doesn't reject that kidney, all right? So this is an antibody-antigen reaction. The same idea it applies to your red blood cells. And I'd like to start with this because everybody can sort of understand this. You relate to that because of transplants. But a blood transfusion is essentially a transplant, right? You're getting somebody else's blood, so you need to be typed properly, okay? Now, when it comes to your red blood cells, there's many proteins on your red blood cells, but for some amazing reason, there's only 3 proteins that matter on your red blood cells in terms of typing, okay? And those proteins are going to be the ones that you display, right? So when you get your blood type, it tells you what proteins that you have, okay? And you're going to reject any blood that doesn't have those proteins, right? So you could get it from somebody else, but those people have to have those same 3 proteins. Otherwise, you're going to reject it. You're going to have antibodies to any of the proteins that you don't have, okay? Hopefully that makes some sense. You won't have antibodies to the proteins you do express, but you will have antibodies to proteins you do not have, okay? And your blood type tells you or tells someone the proteins that you have on your red blood cells, okay? Now, if you don't have those proteins, you're going to reject, if you're given blood with different proteins, you're going to reject it. But we also use these antibodies to type blood, okay? So if we take a sample of your blood and we add an antibody to it, so one of those 3 antibodies, and we add it and we see the blood clump up, then we know that that blood has that protein, all right? Because the antibodies I've added will cause this clumping or agglutination, all right? Now I'm going to go over this again, all right, with different way of looking at it. Okay, so what are our blood types? Okay, so our blood type, there's actually 8 different ones, right? First of all, we talk about the 2 main proteins that most of us relate to, A and B, okay? But don't forget there's the plus and the minus. That's the 3rd protein, all right? So we're talking about the A protein, the B protein, and then the D, or the Rh factor, all right? So here, this slide, we're just talking about the 4 main general groupings. So A, B, AB, and O. So what does that mean? Well, if you have A in your blood type, that means that you are expressing the A antigen. If you have B in your blood type, that means you're expressing the B antigen. If you have both AB, that means that your blood cells, your red blood cells, express both of those proteins, okay? So that means you got 1 protein from one parent and one gene for 1 protein from one parent and 1 from the other, all right? So you're expressing both. But you can also be that person that doesn't have either of those proteins, A or B, in which case you'd be type O. Now, most people and most languages in the world say this is type 0, which makes it easier to understand, which means neither, all right? We say O because it used to be that you say O and 0 was the same thing. Like you gave somebody your phone number, like mine used to be when I was young, 927-5702, right? It means 02, right? So that's where that's coming from, all right? So that O actually means 0. So when you see O, that means it's not having A or B, all right? There is no O protein. All right. Now, so that gives you this picture. Now, if you are type A, all right, in your body, you are producing an antibody to the B protein because that is foreign to you. You don't have the B protein. And if you were given blood that has the B protein on it, your antibodies are going to reject it. You're going to reject that blood, and that's because you have antibodies to B, all right? On the same token, if somebody is type B and they don't have the A protein, they make antibodies to the A protein. So that's the anti-A antibody, all right? Now, if you're type AB, all right, you don't want to make either of those antibodies because then you're going to be rejecting your own blood. So you have neither of those antibodies, okay? But if you're type O, well, then you have a problem, right? Because you don't have either of those proteins. So both of those proteins are going to be foreign to you. So you make both antibodies. You're going to reject blood from A and B, or AB, all right? Because you're going to have those antibodies, because that's foreign to you. You don't naturally express that protein on your red blood cells, okay? So this is the basis of the AB groupings, all right? So if anything I just said doesn't make sense, come see me, all right? Come see me. Now, so when we're blood typing for your ABO status, what they'll do is they'll take an unknown sample, you have untreated blood, and you'll actually add the antibodies, right? So if you're type A and you add the anti-A antibody, you're going to get this clumping effect. So we know that you're A. But if you add the B antibody, nothing is going to happen because you don't have that B protein, all right? So you know type A, right? Conversely, type B is going to be the same thing in reverse. And type AB, you're going to react with both of those antibodies because your red cells express both. And if you're type O, you won't see any reaction, okay? Because that blood sample doesn't have either of those proteins, okay? And this clumping reaction is called agglutination, all right? Now, remember, your blood type isn't just A, B, or O. You have a plus or minus. Okay, so the third protein is the Rh antigen or the D antigen. Okay, you could say Rh factor or D. Doesn't matter to me, but I want you to make sure you know both. Okay, so historically it was called Rh factor, and it's technically called the D antigen. Rh because it was discovered in a rhesus monkey. All right, that's where that Rh is coming from. And the way we express it, instead of saying you have ABD blood, is we say you have a plus or a minus. So if you have a plus, that means you have that third antigen, or a minus means you don't have it, okay? And this is going to be tested exactly the same way. You're going to add an antibody to that protein to your blood sample, okay? And can also, similarly, if you have a plus, okay, that means you won't make antibodies to that protein, otherwise you're going to reject your own blood. But if your blood type has a minus in it, you will make that antibody because it's foreign to you and you're going to reject any blood type that has that protein in it. Okay, so let's look at this, level it up. So remember, there aren't just 4 blood types, there are 8 blood types, okay? So you have your ABO and then a plus or minus for each, okay? So if you're O minus, you don't have any of those 3 antigens, right? If you have none of those 3 antigens, you will make all 3 antibodies. You will reject any type of blood unless it's O minus, okay? Because anybody else is going to give you blood, you're going to have antibodies to it, all right? So someone who is type O minus, they can only receive from O minus. But someone who is O minus is called the universal donor, because since they don't have any of those 3 proteins on their red blood cells, no one's going to reject that blood, all right? So that person is called the universal donor, all right? Now, on the other end of the spectrum is the AB+. That means you have A, it means you have B, and you have the Rh factor, or D, all right? So this person expresses all 3 proteins, okay? Now, this person, since they express all 3 proteins, think about it. What antibodies do they have to these proteins? They don't have any of them, because if they had any one of those antibodies, they would reject that blood, all right? They'd reject their own blood. So this person, this AB positive person, is the universal recipient. They can receive blood from anybody, okay? Because they don't have any of those 3 antibodies, all right? So make sure you understand the difference between the universal donor and the universal recipient, okay? All right. Now, if we look at this and really look at this slide, this should make sense to you, all right? So this is NAB testing using the antibodies for all three, all right? So when it comes to blood typing, all right, there are only 3 possible antigens or proteins to consider: A, B, and D, or Rh. There are only 3 antibodies to consider: A, B, and D, or Rh, all right? The blood type you're given is telling you what antigens are present on that person's blood. So if I am B positive, it tells you that I have both B and Rh or D, okay? What antibodies I have? Well, the antibody that I have is going to be to anything that's left, all right? So this person here who's B positive, the only antibody they're making is to A, all right? Because that's the only protein they don't have, and that's the antibody they're making, all right? So again, the blood type is telling you what proteins are present on those red blood cells that you have to care about. And the blood type then will have all of the antibodies to the proteins not present, okay? Now, when you're typing blood, a positive reaction or clumping reaction will occur with the antibodies added only to those proteins that are present. So for example, if we have someone who has A positive blood, so let's go look at this. The proteins on an A positive person are A and the Rh or D, right? The antibody that person is making is B because they don't have it. Now, when you're typing blood, you're only going to get a positive reaction to the proteins that are there, A and D, and not with the B. So let's go look at that. A positive, anti-A is added, you get a clumping. Anti-B, no. Anti-D, yes, that person's A positive. Okay, so have a good look at this. Go through these blood types and make sure it makes sense to you. Okay. Now this, right, universal donor, type O minus, does not have any of the relevant antigens, so can donate to anybody. Universal recipient doesn't have any of those antibodies, so can receive blood from anybody. Okay. Now this chart, I am never going to ask you something that's complicated on an exam. This chart, as well as this one, is here to help you with your lab homework. In your lab homework, it's going to ask you, identify the universal donor, the universal recipient, and identify who can donate to whom and who can receive from whom, all right? Use these charts. Be smart. Do what I'm telling you. Use these charts. I am not going to ask you a question like this on an exam, all right? Just use it. Do well on your lab homework. There's no point in losing 2 points. Okay, that's how I'm going to grade it. I'm going to take off a quarter point for each thing you get wrong. So that's 2 questions. That's going to be 2 points lost. So just use the chart, okay? Or this one, whichever one you prefer. I like this one. This one's easier to read in my opinion, but whatever floats your boat. All right. Now, the last idea here is Rh factor and pregnancy. All right. So some of my students have gone through this. Now, how does this work? Now, if a woman is Rh negative, all right, so that means her red blood cells don't express that D antigen, right? And if she has children with a man who is Rh positive, the children can, or the child in the womb could be Rh positive. Now remember, this woman is expressing antibodies to Rh, right? Because she's minus. Now, it's not a problem during that first pregnancy, all right? So here you have Rh positive man, fathers a child in an Rh negative woman. The fetus might inherit the Rh positive antigen during the pregnancy, but most commonly in the childbirth, all right? And if you're a woman who's had children, you know it's a messy affair, okay? So there's going to be some exchange of blood between the fetus and the mother. Now, what that's acting as is like a vaccination, all right? So now all of a sudden, she's been exposed to Rh positive, all right? If that fetus is Rh positive. So what she's going to start doing is building up those antibodies. Sorry, that's my phone. Let me just turn that off.
Call from New York.
I apologize, I just hung up on somebody. So what's going to happen here is that she's going to build up antibodies to that Rh antigen over the next few weeks. And this is going to cause a problem if she has a second pregnancy and the fetus is now Rh positive, because she can then use those antibodies that she's built up to reject the fetus, okay? So what they do is that that Rh minus woman is going to be given this stuff, this drug called Rogam. And what it's going to do is sort of sop up those antibodies. And in that way, it's going to allow for the woman not to be activated, right? So it's going to sort of sop up the Rh, all right? And that will sort of protect the fetus. So this is what happens if an Rh minus woman who might have a fetus that's Rh positive, all right? So you'll have to know for the exam what are the combinations. So remember, the combination is Rh negative woman, Rh positive man, and the second pregnancy, Rh positive child. All right, so that's what you need to remember. The last little idea here is blood transfusions. So if you donate blood, whole blood is collected and that can be packed. Whole blood, if you're giving blood, you don't need to receive whole blood. You might just receive platelets or plasma or just different types of cells, right? So this is why they say if you donate blood, you can save all these different people, because they actually very often will fragment the blood. They'll spin it down and they'll separate the red cells and the serum and give people what they need, all right? That's different than a bone marrow transplant, which is very, very cool. So let me explain that. So what's happening here is that the donor is donating stem cells. So what's being used here is to replace the marrow of the recipient whose marrow has been compromised by disease or perhaps chemotherapy. So what's happening is that the stem cells are injected into the patient's blood. Now, in this regard, what has to happen here is that the donor, and this sounds gruesome, but what's happening is that it's taken out of the red marrow. So it's the needle is actually going into the bone. Okay, so this causes a bit of discomfort for the person who's donating the marrow. But that's where the stem cells are. So the desire is to collect the stem cells from the marrow of the donor. Very often it comes from the pelvis. But then when it goes into the patient, now this is the really cool thing, because it can be injected into the blood and the stem cells will always, always, always find the marrow, which is fascinating. And I always wanted to understand how that works. And I did sort of working on that a little bit in my master's. But I still don't quite understand how this works. How does that stem cell know when it's come back to the marrow? So it's come back to its home, essentially. And then it can reestablish the bone marrow in the patient. Now, there's a few different types of bone marrow transplants, and I have them listed for you on the last page of my notes. You don't need to know this for this class, but I sort of list them here. And one's autologous. That's when the patient can save their own stem cells. Okay, allogeneic is when they're given as a match, which is the most common, obviously. The other one is cord blood. Now, the really interesting thing here is that cord blood is really rich in stem cells. And it can be used not just for bone marrow transplants, but later on in life, if there's all kinds of different diseases might benefit from this. So very often, parents will try to bank their child's blood. And there's different ways of doing this, or bank the cord blood. And the desire is to have this just in case the child needs it later on to fight a disease. And there's different kinds of banks. There's public banks and private banks. They work differently. If you bank public, there's little to no cost, but it might be used to help another person as well. Private bank is going to store it. It's going to cost more, but it will store it up just for your child individually. So there's different benefits to that and different ways of doing it. I don't understand all of the politics and logistics of all of it. I just know that there's these different types of banks. Unfortunately, I wasn't able to bank my child's blood, cord blood. I was going to do that, but wasn't able to do it. But anyway, so that gives you a little bit of insight. Now, I hope that I've helped you with this video. Again, if some, especially that antibody stuff, if that's not making sense to you for blood typing, make sure you come see me, all right? Because that's really important. You're going to see it in lab, you're going to see it in lecture, it's going to come up on the exam. And it really is not that difficult. I know when you're first exposed to it, it's like really overwhelming, but it really is not as complicated as you might be thinking, all right? So please come see me, all right? So other than that, I think that's it. And I am just going to stop recording, all right? So thank you very much, and I hope this was helpful.